Yes, thank you, Luboš. Thank you for the invitation. Good afternoon, everyone. I'm going to talk today about the role of the NanoSims to identify and trace microbial processes and interactions. And I choose to show you a couple of applications. I hope I will be in time. From less known work, I would say. So NanoSims had a key role over the past years in solving questions about microbial processes and interactions in a variety of samples. And I can say that it can tackle questions out of research for conventional approaches. I still have fresh in my mind, although it's been a while, the first studies that look at microbial metabolism by the use of isotope tracers and NanoSims, showing very clearly how microbial symbionts can fix atmospheric nitrogen and transfer this to its host. So these studies were extremely important for us in microbiology field because it opened really a huge array of possibilities at that time. So when we are studying microbial processes and interactions, in general, we have three questions: who, what, and how. So we are interested who, so the identity and abundance, so who is out there and what is their metabolic traits, their function in a particular environment, and most importantly, how are they able to perform a certain process, and how is this related with environmental changes. Now, I guess everyone is well informed about now how the NanoSims works. What I want to point out here is how the NanoSims can be used to answer those questions. So you probably already know that the NanoSims is a destructive method, and that we have a source of primary ions which is shooting at the sample, and instead we get secondary electrons as well as secondary ions, and towards this 7 detectors we have that can acquire at different masses, can acquire secondary ions in parallel. We have, based on the secondary ion counts or secondary electron counts, we have basically reconstructing the sample, and we can image in this way, not only quantify, but by image. We usually go with very small fields of view, and here you have an example of a cyanobacteria, a filamentous cyanobacteria with 3 cells, and here it's another one. And you can not only image, as I said, uptake of label, for example, carbon or nitrogen, but you can also quantify it. The most used combination or the most used NanoSims approach is its combination with stable isotope probing and fluorescent in situ hybridization. So stable isotopes are used as tracers of metabolic or microbial metabolism in general, and the advantage of using NanoSims being so sensitive in isotopic and elemental mapping allowed us to actually go in the environment and use the samples directly from the environment in very short incubation times, being sure that using a combination of stable isotopes simultaneously within the same incubation time, being sure to remove the water from the cells and distribute those ideally as single cells homogeneously on a conductive surface. The fluorescent in situ hybridization is necessary to get the identity of the cells because we cannot focus on everything. It's important to focus on a particular target groups, and therefore by using specific molecular probes we can identify either by using correlation with fluorescent microscopy or by putting a particular rare element during this procedure into our cells. So NanoSims analysis, usually it gives us the information on the cells we have in the analysis area. It gives us information on the identity, if that identity it is to this elemental introduction, particularly in a certain group, like here we use fluorine and the specific molecular probe was gamma proteobacteria, and it gives us information on the function on a certain process. In this case, by showing the 13C/12C ratio, it showed us that these cells are capable of taking up 13C labeled glucose. So it tells us something about the metabolism of these cells, the fact that these cells are heterotrophs. So this combinatory approach have been used intensively in the microbial field, and I will point out now also difficulties related with the application of NanoSims, as well as successful examples. So combination of SIP-fish NanoSims can be done, as I mentioned, directly in natural environment in a variety of samples, starting with soil and rhizosphere samples, where you have also plant roots and microbes, as well as soil together, in water and sediment. And as you can guess, most studies have been focused on the water. You have a bit simpler system. You can also do a lot of experiments with technical environments, mixed cultures, for example, or looking at a particular plant root and microbe interaction. It's very important, the step of choosing the stable isotopes as tracers. You need to have a hypothesis and you need to know which type of process you are looking at. And this helps you to choose the label isotope substrates that you will gonna use, as well as the incubation time, for being sure that this substrates will be actually assimilated into the cell biomass, because this is what the NanoSims can measure. The identity and cell abundance is also important, because you are interested to have, first of all, the group of interest to be in a relatively decent abundance that you can analyze in small fields of view, like I showed you. NanoSims can go in up to 80 micrometers fields of view, and it's important you get a decent cells number within this field or area of analysis. The cell identity and function can be done either by the elemental introduction, like I mentioned, the fluorine here, which is not always working perfectly due to some background, but this is true also for fluorescent signal. Sorry. So correlation, I would find it the most difficult part, to really correlate the cells you identified with the within the same area of interest with the NanoSims or during the NanoSims analysis. To know exactly which cells that your target group is actually having an uptake of a particular isotope tracer you use, and that you can quantify by drawing such regions of interest around these cells to particularly calculate the isotopic ratio for these areas. I would say the vital step in all this kind of procedure, it's the sample preparation. And unfortunately, this cannot be, at this point, cannot be made, how to say, systematized in a way that you can use the same sample preparation for all kinds of samples that go to NanoSims. And I will show you here different ways that we prepare our samples before going to the NanoSims. So the most, I would say, simple and straightforward way is through filtration and concentration of cells on polycarbonate filters, which are coated with a conductive layer. It's easily applicable for water samples or small microbial aggregates, and also sediment-free living microbial cells that can be very easily separated from the soil particles or sediment particles. However, if one is interested to look at microbial cells which lives within a tissue or within organic matter aggregates, then you need to consider to embed in a fish-friendly resin because the fluorescence in situ hybridization and mention it's a crucial step for identification and visualization of these single cells prior to the NanoSims analysis. And here you have an example of human adipose tissue that was embedded in a LR white resin and sectioned in thin sections of 300 to 400 nanometers, and a fish was applied on it. And such a sample can be used further for the NanoSims analysis. And this works for soft tissues and microbial aggregates or symbiotic associations. However, more complicated things it's coming when you get soil into the play or sediment, and the sample preparation, it is anyway difficult because you need to ensure not only that the object of your study is growing well, you need to ensure a decent labeling experiment that you are sure that your tracers would be assimilated into the plant and microbial biomass during the time you chosen for the experiments, as well as you need to ensure that you have a subsampling representative samples collected with artifact-free, if possible, sample preparation and collection. And such an example is showed here when you want to look at single cell imaging, microbial imaging in a special context. And I just show you here a drawing of what you have seen before. So the plant roots with soil and microorganisms that suffers the stable isotope probing, so the incubation with tracers for a certain time or at different times even, and after that subsamples are collected at different depths and are embedded in a fish-friendly resin. And after that, this has to be sectioned. And in this paper, we try to find a better way to section. Having soil, there is no way you can section with microtome or ultramicrotome due to the inorganic and particles and minerals. And if you use a regular polishing, what the geologists are using, you are smearing the surface, and it will be very hard to to fish after that and to have a proper analysis. So therefore, we use the water jet cutting to slice, and at the end you end up with such a sample where we apply fish, and we were looking for microorganism of our target group. In this case, we looked particularly for bacteria, and we found such agglomerations that were actually located particularly on minerals. So now this was one step within the analysis. Now the following step is to get information to have a proper characterization of your samples. So we had the fish, that's good. We identified the microbes, but besides microbes, there are much more components there. So what we did, we apply a series of other type of analysis to look and distinguish the root. Like here, you see it with NanoSims. We applied the scanning electron microscopy with EDX elemental mapping to look at elements which are important in general, so micro elements which are important for root growth as well as for microorganisms, and we looked also at the distribution of minerals also in the context of the presence of this microorganism on particular minerals. So correlative imaging, it's again, and you have a characterization. It's important to have before the NanoSims. For some samples, if you do not have this, it's critical, and you can run the NanoSims and then see that you have a bunch of data that you cannot assign to any process taking place in that particular sample. And here, what we did, we look at the we identified the microbial hotspots on the mineral. We look at around these clusters, microbial clusters, to see which elements and minerals we found there, with Raman and electron microscopy. Now, this is probably the most important part, where we go close to the process or a particular role of microorganism to a certain process. Here is the light microscopy images. You see also a section through a longitudinal section through a root, and you have the soil, of course, the microorganism that we found, and we have tried to look at this this area in terms of isotope mapping. So at the end, the most important information for us is to know how much of the label that we initially fed the plant with or added directly to the soil, like CO2, 13 CO2, and nitrate, ended up into the plant roots and into the microbial biomass. And this is the quantification that is unique, and it's extremely important for us to assign a particular process, or assign microbes to a particular process, or to say something about their role in a particular environment. Now things become a bit less complicated if one would look at the root microbe interaction only. However, you need to decide, when you have the bulk roots, what type of primary or secondary roots is actually from this bulk sample. It's important to look at for further isotope analysis by NanoSims and imaging. And that's a difficult step, I would say, one has to take. That's the plant section, and here you can see the magnification. It is the identification of bacteria which is in close tight contact with the root, and we look at the nitrate uptake by this bacteria, as well as the transfer of carbon coming from the CO2 fixation through the plant roots to this microbial cells. And by analyzing this two components, root and different sectors of the root section, as well as the associated microbes, we could quantify the relative assimilation in these two components. Now I would like to go further to the microbial colonizers of plastics. This is also this is unpublished work. And here we were interested to look at how microorganisms, like native microorganisms from soils, will actually colonize plastics, and if these microorganisms that are forming biofilms on plastic are also able to degrade antibiotics. This is in a view or of the contamination we face right now, environmental contamination with plastics as well as antibiotics. So what we did here is to choose two different types of plastics and to set up microcosm experiments, and we set them up to 130 days, so quite a long time, with a label sulfometaxazole, which is an antibiotic. And we follow what happened in time, looking with scanning electron microscopy to see the colonization in time of those plastic surfaces, of course optical microscopy, and then look further to with the NanoSims and fish. So basically this is a schematic drawing that shows you how we had the experimental setup. We had two different kind of experiments. One, that the plastic pieces were directly put in soil, in native soil, which was previously mixed with the antibiotic, and the other one we had the water from above, which was covering the soil, where we had the antibiotic added and the plastic pieces hanging there. So I will show you just preliminary data from the 70 days of incubation. So we have 30 days, 70 days, and 130 days. What we have seen, we have definitely seen differences between different types of plastic, this PE and PS, and we definitely have seen a decrease. So the colonization on those where we added antibiotics, of course, it was much lower versus the plastic pieces where we did not add the antibiotic. I have to say that NanoSims imaging was extremely difficult, not only due to the topography, because these plastic pieces are not perfectly flat, but also because we had a huge array of colonizers and a lot of particles coming from the soil. So what we could see with fish, it's very clear what I told you is that, first of all, you see the differences, this PE, which is higher than PS. This is the colonization on different plastic pieces. And of course, a higher amount of cells we observe on the no SMX plastic pieces versus the one with antibiotics. And the best or the higher community was observed at 70 days, a bit less at 130 days, and very little at 30 days. And the NanoSims data, so this we have a much more analysis done, but I present you just the fish NanoSims quantification, and we have indication at 70 days that we have uptake of this 13C from the antibiotic, from the SMX into the microbial biomass. In blue, you have the control, and in red, you have the so control means cells colonizers without with 12C with 12C SMX. My last story, it's about bacterial fungi interactions, as these are extremely important in soil. It's known that fungi are able to actually transport nutrients and possibly water through soil, and people believes that this ends up in microbial biomass as well, but actually there was a missing evidence that this is really actually happened. So what we did here, we created a setup where we allow the fungi to grow, to overgrown a silicon wafer where we had spores of Bacillus subtilis. And only the fungi that start growing had access to the water, carbon, and nitrogen label sources. And we wanted to see if some of this label is actually ending up in the spores, and how many of these spores will actually bloom into vegetative cells. So this is how it looks, and this is our silicon wafer here that was excised. So that's these are the hyphae of the fungi, and what we have observed with light microscopy is that indeed the Bacillus subtilis spores that were added only on this wafer when it was crossed by the fungi. Showed that they become vegetative cells. That means they have access to nutrients and water, and they start being metabolically active. While the spores further away from this, from the hyphae, actually they stay spores. So then proximity plays a crucial role, and this is important because imaging and quantification, like the NanoSims can do into a special context, which become more and more studied and prove to be important in the interaction. It's extremely important. So here we show that the water it is transported by the fungi to the cells. They become vegetative cells, and only those which are actually aligned very close to the hyphae, but not the spores further away, get access to the water as well as nutrients. And this is the this is an example here of the NanoSims analysis we did, showing the spores further away and the vegetative cells that have access and they grow. So very important, the proximity for the microbial interactions, or at least trophic interactions and exchanges of nutrients and water. At the end of my talk, I hope I did not exceed too much. I would like to point out few challenges I see or we confront with, and one is still the correlation, which is very much sample dependent, and particularly sample identification prior to the NanoSims analysis. Another problem we are confronting with is low isotopic enrichment. So when you have, due to low metabolic activities of cells, and it's harder and harder to convince reviewers that you have actually a process of uptake, like particularly these degradation processes, like you would have a uptake of 13C from the antibiotic, like I showed you. Standardization of the sample preparation and also NanoSims analysis, this is a dream at this moment. So it is very much sample dependent, both analytical conditions as well as sample preparation. And I will add here high cost of the analysis itself and high maintenance cost, kind of that more and more laboratories are kind of pushed to look for users or for payment from I don't know, external users or different other sources that they can survive and continue to do research. And as next steps, and from previous discussions with my colleagues, so there are many people interested to take this technique and apply it further for virus research. At the moment, the lateral resolution of 50 nanometers or even 30 it's not enough or good enough to go into this direction. Well, I heard some rumors that there is an improvement in lateral resolution and will be coming some new instruments with a more enhanced lateral resolution. And one point that I'm always struggling to explain my colleagues and the scientific community, it's interested not only where, in which cell the label substrate ends and how much, but it's interested what the cell is doing with this carbon that was fixed or a carbon that was transferred, which type of metabolites that cell is capable to build up in. And at the moment, imaging and quantifying metabolites, this is really one step forward. Yeah, we know all that we cannot answer with NanoSims at this point, or at least not with NanoSims alone. And with that, I would like to acknowledge my research team, some of my team that participated to the applications I show you are not any longer with me, and to my collaborators that directly participated to the work I show you. And of course, to all the funding agencies, particularly to the DFG for supporting, and of course our institution for supporting financially a lot of the research I showed you. And I would like to thank you, and yes, I'm open for questions or in chat or direct. Thank you.